Hello everyone and welcome to our video on controlling plant growth. This is part of our Module 5 Part 5 series, all about obviously plant and animal responses. What we're going to be doing in this video is having a look at a couple of specification references, 5.1.5c, the experimental evidence for the role of auxins in the control of apical dominance, and 5.1.5d, the experimental evidence for the role of gibberellin in the control of stem elongation and seed germination. Now, it is worth me pointing out here that on your specification, it does point out that you are not required to recall specific experiments. What you are going to have to do is apply your knowledge and understanding of how auxins and gibberellins work, etc., to whatever experiment they could give you on an exam paper. So what we're going to do in this video is actually go through a few examples of experiments. Remember, you don't have to memorize these experiments, but you do need to understand, obviously, how these results led to certain conclusions, because you could be given a similar experiment and asked, obviously, to draw conclusions from it. So we're going to start off with auxins then. Hopefully we remember that auxins are those plant hormones responsible for regulating the growth of our plants. And they're going to be produced at the growing points at the apex of the plant stem. And remember, the apex is the very top bit. What they're going to do is actually cause the plant to grow upwards, and they inhibit the growth of those lateral buds. And that's what we refer to as apical dominance. So when we've got our little plant, what we do want as a plant is obviously more vertical growth, because that means we're growing closer to the good old sun for photosynthesis. What's not very useful is having lots of these little side branches coming off and a much shorter plant. So what we find is the auxins are all to do with inhibiting all of these little lateral buds that we would have that would then become the lateral shoots. So whole idea, promoting apical dominance, inhibiting the growth of those lateral buds. Now, obviously, the whole importance for this ties into photosynthesis, which is also in Module 5. So obviously we can see questions about auxins linked into photosynthesis questions as well. Because if we've got plants growing upwards in that apical dominance, then they're more likely to increase the quantity of light they can absorb on their leaves and therefore carry out photosynthesis at a faster rate. So one of the first experiments that we see here is that when we've actually got our little plant, what they actually did first of all was they decided that they were going to basically cut off the little tip. Now, when they did this, our little plant, rather than having that little tip, it was just a little cut end there. And as a result of that, we then started getting these little lateral buds starting to fall. So when they did that experiment, just cutting the tip off, they hypothesized that this was down to the reduction in auxin in the plant. Obviously, it's a hypothesis, it's a scientific theory. So when we have a hypothesis, we have to test it. So we come up with a new experiment to put that to the test. So the hypothesis is that we've reduced the level of auxin in our plant, therefore lateral buds are growing. How can we test it? Well, all we do is we're going to apply auxin onto that little cut tip, and we see what happens as far as those lateral bud growths go. Now, obviously, you could say that what we get as the result of that experiment is down to exposing those cut ends to oxygen. So because of that exposure to oxygen, maybe the plant is producing something else that causes this lateral bud growth. So we need to then extend our experiment a bit further. We need to do this in a way that is then not going to cause oxygen to come into contact with the inner parts of our plant. So what we did this time, rather than cutting off that tip, is that we actually just apply this little ring of auxin inhibitor below the apex. So what we're doing here is we're just applying a little ring of a chemical there that's going to prevent auxin being transmitted down the plant, and as a result of that, what we see is lateral buds started to grow. So now we've eliminated the potential it's down to that cutting of the tip itself and exposure to oxygen, etc. And we're now coming back to giving us more evidence towards the reduction in auxin is the cause of that increased lateral bud growth. So what we've got then are two theories formed from these couple of experiments. First one, normal auxin levels in our plants inhibits the lateral bud growth, and if we've got a low auxin level, it promotes the lateral bud growth. What we then go on to do is extend this. So remember, when scientists carry out one of their experiments, then they record all their results, they record their method, and they're going to publish that in scientific papers. They end up in journals, and what we're going to do through that process is allow for that peer review. And peer review is where other scientists will obviously read the paper that you've put together, and it gives them the opportunity to then replicate your experiment in their own lab, because everything you've set out should allow them to do this. Obviously, if the scientist can replicate it, that does give credibility to your conclusions. If, however, they try to replicate it using your method and they can't, that does call your evidence into question. So obviously that peer review to confirm your results is important, but it also means that if scientists are reading these papers, they might think about ways to then build on that knowledge and to extend our scientific understanding. And this is what we are now looking at, is those scientists that then read those initial papers and then they went on to develop further experiments, in this case using kidney beans. And from that, we've now got new ideas because we had that initial experiment, gave us that first bit of the puzzle, and now we've built onto it. So ABA inhibits bud growth, and the high levels of auxin keep ABA levels high. So it's not just auxin, it's the fact that we've got the auxin contributing to the amount of ABA that's present in our plant. We also have our cytokinins, and cytokinins promote bud growth. So if cytokinins are directly applied to a bud, then what we see is that apical dominance is overridden. So that means we're going to get lateral buds actually starting to form. If we have high auxin levels, we create a sink for those cytokinins, so they end up going to the apex of the shoot. So if we remove the apex of our shoot, then those cytokinins are able to spread throughout the plant because we no longer have that sink, and as a result of that, what we're seeing is the lateral buds then starting to form. So a couple of different ideas there that came about as the result of finding out that first bit of information about how auxins might inhibit that lateral bud growth, and then working out, is that the sole cause, or is there something more to this story? So the second hormone we need to know about are the gibberellins. And gibberellins, if you remember, are those plant hormones responsible for the control of stem elongation and seed germination. So what we need to do here is put these to the test. If we are making this statement, they control stem elongation and seed germination, let's test and see what happens. So one of the first things that they would have done is to actually test this gibberellic acid on a range of different plants. And what they found was that when we apply the gibberellic acid to dwarf varieties of the plants, they grew taller. So what we can see here is that gibberellic acid is likely to be responsible for that stem growth. And obviously this is just quite a straightforward experiment that we can do of just having our plants, giving some of them obviously our test substance, in this case the gibberellic acid, and then recording the results. Now obviously this does bring into the kind of questions where we could see practical based questioning, and therefore we could be seeing things to do with control variables coming in, etc. So just think about all those practical skills that in theory you have done in class. You should have done obviously 12 practicals as a minimum across your A-level course, some of which are obviously looking at these control variables. Now if we're thinking about plants, then we need to be referring to using the same species of plant. Let's avoid saying use the same plant, because that's a little bit nonsensical, because if you were to use just the same plant, that basically tells us we are using one plant for every experiment. So if we've got a dwarf variety and we've applied gibberellic acid to it, then it's grown taller. How are you then going to do something with that same plant in the second experiment? doesn't work. However, if we are using the same species of plant, then that's going to be better. Obviously, we could say we could use clones of that one plant because then they're all genetically identical to each other. But same species of plant is the best way to phrase that one. Obviously, other control variables to bear in mind where we're talking about plants is things like the volume of water. Please get out of the habit of using the word amount full stop on your papers. Always refer to volumes or masses, numbers. Do not say amount because amount not really getting your marks on an A level. And then think about some of those more basics, okay? Times, temperatures, concentrations, and obviously the volumes. Those are going to give you the main kind of headings that you can use for pretty much any experiment for those control variables. And then in this case, if we wanted to extend this further than just gibberellic acid causes them to grow taller, we could actually do different concentrations of gibberellic acid. And that links in to serial dilution that you should understand. So the second aspect that the gibberellins have some kind of control over is obviously the stem elongation. And the way they cause this growth is actually in these regions called the internodes. When you're looking at a little plant, I've given you a diagram here to hopefully make it a bit clearer, then the nodes are pretty much where you get the branches or the leaves coming off. And the internode is the region between two nodes basically. And what we find there is that the gibberellins actually cause that cell elongation and cell division in that internode region, which is then what causes the stem to get longer. The other aspect of our gibberellins is, of course, the breaking of dormancy. And hopefully we all know that seeds are a dormant form of a plant. So that's basically how the plant is able to reproduce and not die across the unfavourable growing seasons, because the seeds are in a dormant state and they will only come out of that dormant state, in theory, when the conditions are adequate for growth. So when we need those seeds to grow, we've got to break this dormancy. And when seeds actually absorb water, and obviously they're only going to be doing this when the conditions are more favourable, i.e. the ground isn't frozen, etc., then the embryo is going to release gibberellin. So what we can see here is that it's that process of absorbing water by the seed that leads to the gibberellin being produced. The gibberellin is then going to travel into the alurone layer in the endosperm region of our seed, and it's there that it's able to produce amylase. And hopefully remember that amylase is an enzyme, we know that because it ends in ASE, and that is going to obviously be able to break down the starch grains that are stored within the seed. That means that we've then got the sugars available for respiration to then provide that ATP necessary for all of those processes for our plant to undergo growth. In terms of experiments proving this seed dormancy breaking ability of our gibberellins, then what we actually use are a certain type of plant. So here we've got it, the Aradiposis, and we use a mutant strain of it. And basically this mutant strain doesn't produce its own gibberellins. So quite simply, all we need to do is have our little seeds of our mutant plant, and they will not actually break dormancy on their own because they're not producing their own gibberellins. If we spray them with gibberellins, they will germinate. So it's a nice simple experiment there that we could do. Obviously this could link into changing concentrations of gibberellins to see the effect, and obviously in that case think serial dilution, think about how we could record that accurately in terms of counting the number of seeds that have germinated in a certain time frame, calculate a rate, etc. There's various little mathematical bits that could come in here, but we've got separate videos about the maths in a later video. As always, I hope you found this video useful, and don't forget to subscribe to the channel so you can next see when a video is uploaded. And of course, head on over to the website where you can see other resources to help you in your A-level studies.